So, welcome everybody. Thank you for being here and for the day 2 of the week. I'm pleased to see everyone and so much of participants for our event. So, first of all, I am Corentin Lignac, the general student chair of the student branch, IEEE student branch of Bordeaux, of the University of Bordeaux. So I will let Aurélien introduce your first talk, which is linked to the EMB chapter. And then we will have a small coffee break at the outside of the laboratory. And then we will have 2 talks from EM chapter, so instrumentation and measure chapter. So thank you and have a good talk.
Hello everyone. We are really pleased that we can now have a talk in person, and so we are really pleased to welcome Professor Michela Chiappalone here in the IMS. So I am Aurélien Way, the current chair of EMB chapter of the B branch. So EMB chapter is the Engineering in Medicine and Biology Society of IEEE Association. Its field of interest is the development and application of engineering concepts and methods to biology and healthcare to provide effective solutions for health problems and medical application. So I will introduce our today's lecturer. She obtained a PhD in electronic engineering and computer science from University of Genova, so in Italy, in 2003. She then obtained a postdoc position first at the University of Genova and then at the Istituto Italiano di Tecnologia, so IIT. Then in 2013, she got a researcher position in the same institute. Since March 2021, she has been appointed as Associate Professor of Bioengineering at UNIGE. And the same year, she has been named IEEE Distinguished Lecturer. She authored 76 papers published in international journals, more than 60 peer-reviewed contributions to international conferences, and she's also the editor of 2 books. So now I give the floor to you, Mrs. Chiappalone.
Thank you very much. I hope you can hear me well, even if I have this mask. So first of all, I wanted to thank you, the B branch, for this kind invitation, because actually it is the second talk that I give at present. The first one I was invited by Danielle, who is here. So I'm very happy because I could travel again after a long time. So the title of my talk today is Neuroengineering Solution for Restoring Brain Function. And I will tell you something about the research activities that I've been performing in the last, let's say, 15 years, more or less. Maybe you don't know that brain disorders affect more than 1 billion people worldwide. And actually, this number is likely to increase in the next year due to the aging of the population. If we want to quantify the impact that brain disorders have on the population, we can use this metric that is called DALY, which stands for Disability Adjusted Life Years, which are the number of years that a person loses because of a disability. As you can notice, the DALY is the highest for neurological disorders. It means that it's the necessity to find solutions for treating this type of pathologies. Actually, pharmacology, which is largely used in medicine, is not the best suited solution for this type of disorders. Why? First, because pharma companies have not found specific drugs, innovative molecules in the last years, which are targeting nervous system. And second, because whenever a neuronal pathway is broken, pharmacology maybe is not the best treatment. And if you see the process that brings from testing compounds up to an FDA drug, you see that the process is highly time-consuming, is very costly, and you know that pharma has a lot of side effects. So we need to find alternative solutions. And actually, this solution can be provided by neuroengineering, which I like to define as a tale of two loops. From one side, there is neuroscience, which poses questions, and technology, which poses solution to this question. But there is also another part of the story. Technology can provide new devices that can interact with the brain. In order to develop fantastic new technologies like brain-machine interfaces, neuroprosthetics, or electroceutical solutions, it means that we can use electrical stimulation in order to treat specific brain pathologies. And it is exactly this field, the one in which I'm working, with the final goal to target new technologies to treat diseases that affect people. And I like to call them the personalized solutions because they must be tuned on the patient features. So how to reach these actually ambitious goals? I can say that there could be two possible ways to face this challenge. From one side, we can develop new devices that are going to replace part of the damaged tissue. So technology can be used as a substitute of the dysfunctional brain. And from the other side, we can provide devices that help the brain to help itself. So basically, we are exploiting what is already there, but we can tell the brain to do it better. And how, the way in which we can do it is from one side to develop technologies that can bidirectionally interact with the brain or with the nervous system in general. And from the other side, we can design a personalized neuromodulation solution that goes exactly in the direction to promote plasticity, which is already in the brain, but maybe is not functioning in the correct way. So there are two main questions I would like to answer. The first is how we can exploit advanced technologies to develop innovative procedures for the replace part. And for the retrain, the question is personalized neuromodulation strategy able to promote an effective recovery? So in order to respond to this question, I use what I like to define a translational methodology. It is a typical engineering approach where you use reduced model in order to try to respond to basic question, and then you can try to answer the same question in more difficult and more complex system. And I use biology to do that. So I use reduced experimental model in order to test technologies and algorithms. And then starting from simple system, I can go up and testing them in more complex brain system like in vivo system and targeting neurons. And the basic starting point is these things that I'm pointing at. These are in vitro cultures of neurons, so biological neurons that can be grown over devices which are called microlateral arrays. And these simple devices and systems can be used in order to respond to simple questions. This idea is not new. I like also to mention Professor Potter, who first designed, let's say, this type of approach, and was a pioneer in using this type of system. And as you reported here, this simple in vitro network provides an excellent test bed for testing hypotheses and studying plasticity, and also interfacing modalities. So what I will present you is the first part in which I need to characterize this simple system from the point of view of the signal that is exhibiting, then design possible interfacing modalities, and all these bring to the neuroengineering solution. So let's start from natural characterization. So what is the experimental model that we have been using? We start from neurons coming from embryonic rats. These are of animal origin, but we are now moving towards more ethical approved systems which are based on human-induced pluripotent stem cells. You can take a piece of skin from each of us, and you can basically transform them by a not so easy process into neurons, so they can be converted into neurons. So you can have neurons that are belonging to a specific patient, to a specific subject. So these avoid also experiments on animals. But in general, what has been done in the past year is to use these neurons and grow them over the surface of substrate-embedded microelectrode arrays. This is a very standard one. There are 60 electrodes. This dot, the black dot that you see here, are electrodes and are able to record the electrical activity from this set of cells. And what I've been doing in the course of the years, I was especially interested in understanding the type of signals that we can record from each of these electrodes, to understand the natural behavior and to be able to characterize the specific patterns of activity that are exhibited by this culture. Notice that these type of patterns are not so much different from what you find in the brain of each of us. That's why it's important to have these simple models where we can test algorithm and technology, because the translation is quite easier then. And just to provide you an overview of what we can do with this simple system, which has been, by the way, recognized as a fantastic model of neural dynamics, you can use the same electrode that you use for recording also for simulation. And you can perturb the network by stimulating it. You can treat the culture by putting some drugs in there. And you can also change the topology of the network by imposing a specific organization of the neurons. And in turn, when you modify the topology, as a consequence, you modify the dynamics. So you modify the neural activity of the culture. And if you are interested, I suggest this book that I edited, which is a sort of collection of the very latest results obtained in this field in the last, let's say, 5 to 6 years. So it's very nice to provide an overview on every single part, from the cultural modalities up to the interfacing the systems. So going much into the details, the type of signals that we can see, each single square here represents 1 second of electrophysiological activity from one electrode. This gives you an overview of 1 second of activity of all the electrodes of the array. We can zoom. This peculiar pattern is called bars, which is a sequence of activity, which are the spikes, which are related to the action potential that are fired by the neurons. And another way to represent this type of activity is the so-called raster plot. I don't know how many of you are familiar with these things, but on the x-axis you have time, on the y-axis you have identification of the electrode, and with this plot you can have quite a nice overview of the activity of all the electrodes at the same time, and for also longer time scale. And there are basically 2 things that you can easily understand when you look at the raster plot. The first is that more or less all the channels are behaving in the same way, because these networks are highly connected. And the second, that there is a pattern of activity that emerges only when you look at the culture as a whole, and this is a highly synchronized event on all the channels. I forgot to tell you that every single dot here represents a spike. Okay? So when you see some small groups, these are bars, and when you see bars on the different channels, you have a pattern that is called network bars. So this type of pattern is peculiar of the culture, of this type of culture, and it is a sign of healthy of the culture itself. I wanted to explain this to you because it will be used then for another part of the talk. And yes, so you can also perturb the network by stimulation. You can use one of the electrodes to deliver stimulation, and you can get, as a response, some evoked spikes or even an evoked burst. And if you have a sequence of stimuli and you put the trial by trial, one over the other, the response to the stimulation, you can build up what is called the post-stimulus type histogram, which tells you somehow the probability of evoking a response given that there was a stimulation. And I introduced this to you because, again, I will use it later. So, okay. So just to conclude this part of network characterization, I hope that this is clear that this culture have electrophysiological activity. Network burst is a peculiar feature, and this culture responds to electrical stimulation. So now, focusing on the primary goal, the one of the replacing value. Okay. We actually faced this problem by proposing a European grant, which was called Rainbow. And the goal of this European project, which I had the luck to coordinate, was exactly to provide technological tools to design the next generation of neural prosthetics. And the idea was, in fact, to test whether this technology can be used to substitute the functionality of a simple in vitro system by using a reduced model of the brain. Okay? And the project was conducted by myself and my great partners, Paolo Massobio in Genova, Paolo Bonifazzi at the time was in Tel Aviv, and Timothée Levy and Yannick Bornat is here in Montpellier. And these are the main, let's say, the main philosophy behind the project. So we have been starting from simple in vitro system. As you can see, this culture is different from the one that I showed you before. There are basically two populations that are connected via neurites filaments, which are part of the neurons, that can go through this communication channel. So basically, you have two subpopulations, okay? And one damage that we were able to do was to cut these connections and replacing them by exploiting what we called a bidirectional bridging approach. We were identifying a specific pattern of activity in the red population, and every time that we identified, in particular, an actual burst, we were delivering a stimulation in the blue one, and vice versa. In this way, we were able to artificially reconnect the two populations that were not anymore connected in a natural way. And the second approach was to think about one of the two populations was dead, so we wanted to completely replace it. And the idea was to replace it by using a spiking neural network that was inspired, let's say, was mimicking actually the behavior of the missing population, and again allowed this artificial connection between the remaining module of the neuronal system. And this was published a couple of years ago in the journal Science by Cell Press. I will go much more into the details of this technology and of the results. So first, we were able to build up this culture, which was peculiar. We put some biocompatible mask over the surface of the array. The cells were growing only where there was the opening in this mask. And the cells were able to spontaneously exhibit electrophysiological activity. Then we performed the first set of experiments. What you see here is spontaneous activity. So these neurons are able to spontaneously exhibit electrical activity. And what you see here is a raster plot. I will now start a movie. This is the raster plot from the blue module, the raster plot from the red module. And this is simply a spike count, which is called the instantaneous spike rate, and that you see here on the top. So what we monitored was simply the spontaneous activity of these two modules over a 4-hour period. And if I start the movie, you can see that more or less the activity does not change in this short time frame, but of course we were also able to quantify by counting the firing rate, so the number of spikes per second over the 4 hours, which did not change. This is a parameter that we made up is called the probability of single module network burst. It means that it evaluates whether the probability of generating a network burst comes from a single module or from two modules. Specifically, this is from the single module. It is very low because actually this network burst is involving both modules at the same time. Okay, and we also computed the cross correlation between the top and the bottom population, which was staying high for all the time of our analysis. This is a control experiment to test whether everything was working well. Okay, then we performed a damage. Which by means of a laser that was able to physically cut the connection between the two populations. And the second type of experiment was, again, a control experiment with the lesion, in which we have spontaneous activity, the lesion, and spontaneous post-lesion activity. I think it is clear for everybody that when the movie starts, you see a difference with respect to what we obtained before. Because now we don't have that synchronous network burst in all the module, but we have network burst which is mostly confined either in the blue or in the large module. The quantification says that the culture is healthy because the firing rate was more or less the same. But look, the probability of single module network burst that was low at the beginning goes up and reaching almost 1. And in fact, there is the high probability to have a network burst which is involving just one module. And the cross correlation is high only at the beginning, as well as the cross correlation. So we have a nice model, simple. Of course, it's not. I'm not saying that this is what happens in the brain, but at least we can test our hypothesis with that. And this is the work of Yannick and Timothée, in which they designed an entire all-hardware closed-loop system in order to perform real-time processing of the data that are coming from the biological culture by performing real-time spike detection, real-time identification of the pattern that is called the network burst, which is a peculiar feature of our culture, and this is the one that actually is used as a trigger for stimulation. Then the stimulation can either go back to the culture itself or go through a spiking neural network, and which is the same depending on the type of parameter that we are using. Then the network burst detection is detected within the spiking neural network and goes back to the stimulation. The spiking neural network is tuned according to the features of the culture that we are recording, okay, in order to meet the same type of network burst dynamics that we saw in biological culture. And here are the results for the bidirectional bridging approach. We have the spontaneous activity at the beginning, the lesion here, the spontaneous post-lesion, the bidirectional bridging, in which the whole hardware system I'm using, and again the spontaneous. I don't have video for this, unfortunately, but you can see that before the lesion, same situation as before. After the lesion, disconnection. During the bidirectional bridging, again, we see that at least qualitatively we have a similar condition like the pre-lesion one. And when we stop this bidirectional communication, we go back to the post-lesion condition. And these have been quantified. Look at the cross-correlation here, and also the probability of single module network burst, which goes back to values that are similar to the spontaneous pre-lesion condition. So the cross-correlation is partially restored, and the probability that of the single module network burst is comparable to the pre-lesion condition. And similar results have been obtained with the second paradigm, the hybrid bidirectional bridging, same type of protocol, but this is second paradigm in between, in which we have the situation at the beginning, no lesion. During lesion, the spiking neural network here was doing whatever it wants. Only during the hybrid bidirectional bridging there is a synchronization of the activity from the biological and the artificial one. And when we stop the hybrid bidirectional bridging, the situation comes back to the pre-lesion condition, to the post-lesion condition, sorry. So again, if we quantify the cross-correlation area, improves, is not fantastic, but at least is better than during the post-lesion. And the probability of single module network bars, again, reaches values that are similar to the pre-lesion. So what are the main take-home messages for this part? For sure that in vitro platform can be used as a test bed for testing technologies and algorithms. Why we are doing this in hardware? Because it's closer to possible implantable devices for the future. And this approach can be also exploited, we are doing it now, for in vivo application. Okay? Another possibility that is provided is also to exploit, for instance, neuromorphic computing to make a computation. Like recently, we have been collaborating with a Spanish group, and we tested their neuromorphic-based device to detect the burst in the culture without performing any real-time closed-loop processing, but at least it was a proof of principle. And also, there is, if you're interested, there is this review, which both myself and Timothy have been invited to participate, in which there is a nice review of all the works in which a biological system has been put in contact, in bidirectional contact, with an artificial one, to study, to investigate adaptation and plasticity. This type of system in which a biological element talk in a bidirectional way with an artificial one is called a biohybrid system. And this type of system is the one that can be exploited to test technological and scientific questions. Okay, let's move to the training part. So now the focus is on animal model. Why? Because if you want to develop therapeutical solution, we needed to exploit the circuitry that is within the brain. And if you want to promote plasticity, you have to use this kind of system. But at least you reduce the number of experiments, because you can learn something from reduced experiments that you have done in vitro. Okay? So this is a different set of experiments. We did not yet use the device that you saw before. Now the question is a little bit different, because the idea is to develop a neuromodulation strategy that can improve the performance after that there was a lesion. So this work is rooted on a work from a group that comes from, that is located in the United States. And the main, let's say, underlying principle regarding plasticity is, let's say, summarizing this sentence: cells that fire together work together. It means this is related to a seminal work by Jackson and Fats in a famous paper that was published in Nature in 2006, in which they demonstrated that by artificially pairing two neurons within the brain, they were able to potentiate the connectivity between these two neurons. So, and recently, more recently, this group in Kansas City, Randy Muda, David Kugelmoss, with whom we had an intensive collaboration, they published this fantastic work that was published in NAS in 2013, and I'm going to briefly explain the experimental procedure. So what they did, this is a schematics of the brain in which you have the sensory cortex, the motor cortex, and the premotor cortex. The rat is performing a task which is called the pellet retrieval task. So it is going to use his forelimb in order to retrieve a pellet of food through a small opening. Okay, and the type of well, to schematize this, you see the somatosensory cortex, motor cortex, premotor cortex, and they go down to the spinal cord and to the muscle. What had been done from the experimental point of view, they were able to focus, impair the motor cortex to create a local damage in order to impede the rodent to perform the task. Again, this was to, let's say, model what happens in humans when they are affected by stroke or traumatic brain injury. And in the end, the motor cortex was not able anymore to deliver signals down to the spinal cord. So what happened, they provided an artificial pairing between the premotor cortex that still have downgoing signals to the spinal cord and the sensory cortex in order to develop what they call the brain-machine-brain interface. So every time that premotor cortex was firing a spike, the stimulation was delivered in the sensory cortex. And this pairing was creating a connectivity between these two areas. And in fact, I have the results here. In control condition, the animal, let's say, can also have spontaneous recovery. If you don't do anything, there is some kind of recovery, also in human factors. But in control condition, after 20 days, after the lesion, there was not much improvement in the behavioral outcomes. This is the recent success. So the success rate of performing well the task in a healthy animal is around 80%. So in control condition, in which you don't perform any type of stimulation, it remains around 20%. There is not much improvement. They also perform what they call an open loop stimulation. So they perform a stimulation in the sensory area here, and that's it, without any pairing to other parts of the brain. And you see there is some kind of improvement, but it doesn't go up to the 50–60%. When they were able to perform this artificial pairing, in which stimulation was driven by the activity of the other area, there was a great improvement reaching values that were very much similar to the pre-lesion condition. Okay? So this demonstrates that at least from the behavioral point of view, they were able to reconstruct again the behavioral capability that they had before the lesion. So why we are interested in this type of activity? Because actually what can be noticed in this experiment is that every time that the stimulation stops, the rat becomes to the condition before. So there is no retaining, there is no retention of the performances. So this is an indication that maybe something in the design should be changed. And how to change is provided by investigating better what happens at the level of the electrophysiological activity of these networks. So that's why we were actually interested in better understanding and going into the details of the electrophysiological activity exhibited by the networks that are involved in this artificial pairing. And this is part of a project that has been funded to promote bilateral activity between Italy and the United States, mostly related to the activity of my former PhD student, Alberto, that you see here. And actually, Kansas City was fantastic because there is nothing there, so you have here time for work and nothing, no distractions, let's say. We performed actually 4 experimental campaigns in which we wanted to study the effect of this closed loop stimulation in the healthy anesthetized animal to check what the effect of this type of open versus closed loop stimulation has in terms of electrophysiological pattern. The same type was then studied in healthy chronic animals, so behaving animals. And more recently, we started experimental campaign also in injured animals, both in anesthetized and in the chronically implanted condition. All this is necessary, requires a lot of expertise from the experimental point of view, because these type of experiments are quite highly time consuming, but also a lot of expertise in data analysis and signal processing. And this is something on which we are also working a lot. This is a freely available software that allows to manage this type of experiment that was jointly developed by Federico, my PhD student, and Max, a PhD student in the Ranking Noodles lab. So let me explain this type of activity. I know this is quite close to neuroscientific studies, but I think it has a lot of implication also from the neuroengineering point of view. So this is the interstimulus interval distribution during the activity-dependent stimulation, so the closed-loop stimulation. And this is one of the random stimulation, which is the one that is delivered in an open-loop modality. In these specific experiments, we targeted also two different stimulation areas in the sensory area, the forelimb and the paralimb. This is not so much important. Just keep in mind that we were replicating the same type of experimental paradigm that you saw in the experiments before, but now looking in more details to what happens to the electrophysiological signals. And we were mostly focusing on the spiking activity here. So I think the more interesting results happen when we perform stimulation. The experimental protocol presents some spontaneous activity recording and then 3 consecutive sequences of stimulation, either in an open loop or in a closed loop approach, which is open loop random stimulation, closed loop activity dependent stimulation. These are post-stimulus time histogram, and the quantification of the post-stimulus time histogram is provided by the area below this histogram, which tells you how many spikes are on average generated by this type of stimulation. I think you can appreciate that these 2 blocks of 3 box plots here, which are the one in which activity-dependent stimulation, so the closed-loop modality, is delivered, are much higher than those produced by the open-loop stimulation. There are 4 blocks because I said that there are 2 different areas in the somatosensory cortex, but more or less they behave the same. So the take-home message here is that the activity-dependent stimulation, so the closed-loop modality, is able to promote a better response in the network with respect to the random stimulation. And the same results was obtained by performing experiments in the chronically implanted animals over a time frame of 21 consecutive days, in which every day the rat was receiving the therapeutical stimulation over this time frame. And we obtained more or less the same results, in which we obtained that the activity-dependent stimulation was better in training network activity with respect to the open-loop random stimulation. And yeah, these were published very recently. This one in cerebral cortex, the other as well. And we are now focusing on what happens in the lesion event. But there is also improvement that we are also facing in terms of technological development, because as you can imagine, the timing of the stimulation is very much important. So in order to record and stimulate, we are using this commercial system by Inton Technologies, which relies on an FPGA in which spike detection is designed. But the problem is that their spike detection is not so reliable. So the work that was done by Stefan and Max was actually to redesign spike detection, also with the help of Yannick. And that was able to basically identify in a quite good manner the shape of spikes that are coming from a single neuron. And this is done by performing a set of checks and control and comparison on different features of the spikes and comparing to some threshold values. So this is a technological improvement that allows also to have experiments more reliable from the point of view of identification of the spikes during the closed loop stimulation. Okay, so I'm almost done. Regarding the retraining, we also demonstrated in this case that the closed loop is more effective than open loop stimulation, but further investigation are necessary in order to basically also understand what happens in the injured animal, and this is what we are doing right now. It is also important from the technological point of view to improve the real-time processing of neural signals. We have been focusing on spikes, but also local field potential, which are the low-frequency signals that you can also read, for instance, in humans. Mostly with an EEG, you can read the low-frequency signals, can be used to improve the performance of this closed-loop approach. And also, another thing on which we are working is that we can explore also new neuromodulation strategy. we understood that the closed loop is functioning very well, but this does not mean that the open loop is bad. Because, for instance, for translation in humans, especially by using non-invasive brain stimulation approach, it could be better to use an open loop approach, because the closed loop is very difficult to implement with non-invasive brain stimulation now. So we are also investigating alternative open loop approaches which are more personalized in order to better entrain activity and obtain, if possible, similar results to the closed loop. And in particular, the part of activity in which technological improvements is necessary is done in collaboration with Matrix, and to the work of Mattia, another student that will start a PhD program in the next month. So what's next? Of course, the target is also humans, but for the moment we are still working at the preclinical level. This is exactly what we are doing now, thanks to the work of Marta. The idea is exactly the same that I anticipated before, providing therapeutical neuromodulation, so developing open loop stimulation modalities which are personalized, so which are tuned to the specific features of the network that we are recording from. And this is done in collaboration with neurologists of the neurological hospital in Italy and my former affiliation of IIT, thanks to this joint. Another work that we are doing is we have a small project with Timothy Levy working here at IMS, in which the idea is to exploit the spatial neural network in order to provide a novel stimulation modality for the in vivo animal models. So the idea is not to provide, let's say, an open loop stimulation which is, let's say, decided or tuned by mathematical model, but it is an emerging properties of the SNN, which is then connected back to the animal. No closed loop here, but simply a stimulation that is driven by a spatial neural network. And this is the idea. So we have started performing experiments. They were building the model, and the model will be used to stimulate the animal itself. So these have been just started, and we hope I will be here, by the way, in a month to do the first some first work with Timothy. And this is another project that I want to mention because also Sylvie Renaud and Yannick are involved. This is the work of this guy, which has been just approved by European Commission as a Marie Curie individual fellowship. Is again on closed loop neuromodulation, taking into account also the phases of sleep, because we know that during sleep there is memory formation and memory retention. So the idea is to provide stimulation in specific window in which plasticity is maximized. And in order to do this project, there are a lot of competencies that are coming around, competencies on closed loop system that we can find here in Bordeaux, on experiments in Genova, and also competency in business and in the possible exploitation thanks to the collaboration with this company in brain and neuroelectronics, which is based in Spain. So unfortunately, it has not started yet because the issue is from Brazil, and Brazil has border closed till now, but I hope we can start not so far away from now. Okay, this is the final slide. I think it's the most important because I would like to thank you all the students that have been working with me and all my collaborators, either in Italy or outside Italy. And yeah, I don't know if I talk too much, but I would like to thank you very much for your attention.
So thank you very much for your very interesting lecture. So now, as a question to ask for lecture. On YouTube. Okay. So I will start. I was wondering, you told us about specified open loop stimulation And I was wondering if it was specified on the special zone of the brain, or a special area of the brain, or more specific. It could be also specified on the patient.
Yeah, okay, yeah, it's clear. Regarding the personalization of the stimulation in the animal model, the idea is to take the activity from the premotor cortex, that is the one that is used also for the closed loop in order to trigger the activity in the sensory area. So we are analyzing the electrophysiological signals there and taking some peculiar features like the firing rate, like the timing of the spikes, reproducing the inter-spike interval distribution. So this is also a means of personalization. But this you can do where you can record the spikes. The point that you observed is very important. In humans, how it is possible? It is possible because there was a paper that was published a couple of years ago in Journal of Neuroscience, in which the authors basically tuned features of the stimulation that was delivered in terms of transcranial alternate current stimulation. Typically, the TACS is delivered at a fixed frequency. What the author did was to deliver a stimulation in which there was not tuned on a specific frequency, but was ranging in frequency that were collected by pre-analysis of resting state activity through EEG recording, and have, let's say, a more broader distribution of the stimulation frequency according to what was specific on the patient. So they developed this technology, which they call the individualized stimulation. And so, I mean, our idea of personalization was exactly to go in this direction, because they demonstrated that with the individualized stimulation, they had better motor responses and the better network and training. So this can be done also in a non-invasive way. That's why they are interested in those modalities, because they are more translational.
Other questions?
Don't be afraid to put your question.
Yeah.
It will be your comments to discuss with Michela.
I'm here in anything.
And maybe you'll be able to talk with her after. Même si vous voulez la poser en français, pas de souci, on la traduira pour vous. Si vraiment. But yeah, don't be afraid. If you have not any more questions, we just
I want a question. I think that I guess that it's a long process, but how long does it take to be able to be authorized to do clinical tests on human beings after you in vitro or in vivo solution?
I'm not very familiar with clinical trials in humans. I mean, I've been doing that while I was in IIT, but for other types of experiments, for instance, it also depends on what you want to do. Because if you want to perform, let's say, study with EEG and electrical stimulation non-invasively, that is quite easy. It takes a few months to get approved. If you want to perform, for instance, experiments with the robotic exoskeleton, if they are not CE approved, you have to go to a different procedure. And again, it takes a few months. If you want to use more invasive technology, I don't have experience, but I guess it will last a lot because invasive technologies can have higher side effects. So there is a longer procedure for doing that. But for standard, let's say, non-invasive approaches, I think it's few months. And if you don't use anything new, new technologies, it's quite straightforward. If you develop something new and you want to test, again, it takes more
time. Why is there any other research done in the direction of rebuilding brain tissue without replacing it with electronics?
Yes, there are. For instance, by using stem cell transplantation, this is purely biological approach. I think you were meaning that. There are, I'm not very familiar because again, it's more biologically oriented, but I know that I know a paper that was published a few years ago in which a group in the US was transplanting, let's say, stem cells into a patient with no, it was an animal model, of course. It was an animal model for epileptic seizures, and they demonstrated that by transplanting these, let's say, non-cytological cells, they were able to diminish the number of seizures for epilepsy. So it's something that is, let's say, on their way. I'm not so aware, also because at the beginning was not an easy procedure, because integrating, let's say, an external, it's called graft, into the system is not so easy from the point of view. It can be rejected, basically. So we think that this approach, it's somehow, even if it does not appear like that, it's faster. Yes.
So you were speaking about the rejection. Are there studies on new technologies that will manage to make this rejection disappear, to beyond rejection?
I don't know exactly. I mean, I'm not familiar with that, but well, there are, let's say, two types of rejection. Maybe is not the correct word, but for instance, they can create like local tumor. Okay. There is also a process, an inflammatory process that comes every time that you make also an implant, a chronic implant, because the immune system attack the device, like also the electrode, and create a sort of barrier around it, and this also impede the good quality of the system. So there are technologies that are aiming at reducing this type of attack from the inflammatory system. This is more for material scientists, which are developing new technologies aiming at better interfacing with the brain tissue and in general with biological tissue.
Thank you for your questions. I think it was very interesting, and I would like to thank you again, Professor Michela Chiappalone, for this talk. I hope you enjoy it, and I would like to say a special thank for Aurélien, which is an undergraduate student like you. So he did all by himself the introduction and the conclusion of the talk. So thank you to him. Basically now it's time for a small coffee break during approximately 15 minutes. So I will invite you to go outside to the cafeteria. You can see there is a buffet. You can participate and talk with the PhD students that are here, and also with the professor that is invited also to the coffee break. So thank you all for coming. It's a real pleasure to have this you people.
Thank you.
Have a good Oh, and there are two more talk just after the copyright.
Thank you
so you can come back here. It will be online to you. We will broadcast it here.
I understand it's quite far away from what you're used to, but I hope it was interesting for the students.
Not very